Hi, my name is David Benjamin, and I'm a grad student in the Heinz Lab at the Koch Institute here at MIT. And I work on using zebrafish to model metastasis. Metastasis is when tumor cells leave a primary tumor, which is where cancer originates and spread to other organs throughout the body. Metastasis is the overwhelming cause of cancer mortality. So one gene that our lab has been interested in for quite a while now is called YAP. And YAP is a really nasty gene. So YAP has been shown to be involved in numerous cancer types and promotes growth and metastasis in all of them. And what YAP actually does is it's a transcriptional co-activator. So what that means is that in cooperation with other genes, it turns genes on and off. And what I'm trying to figure out is actually which of the genes that YAP turns on and off are causing metastasis, because YAP regulates quite a few genes. So I'm injecting tumor cells that I've made to overexpress YAP into 2-day-old zebrafish embryos. And then because the embryos are transparent and develop outside the mother, I can follow these tumor cells over the course of hours to days, and I can really get nice movies showing how YAP changes the behavior of the tumor cells in circulation and then also at the metastatic site. So in this image you see red blood vessels because this is actually a genetically modified fish that expresses a red fluorescent protein in all of its blood vessels. And then the green are tumor cell nuclei because I've also modified the tumor cells to express a green fluorescent protein in the nucleus. So I've been mainly working with melanoma cell lines, and melanoma is the most deadly type of skin cancer. And this cell line here is actually a colon cancer cell line. And so in this experiment I was trying to see if the results I saw in melanoma also hold up in colon cancer. So generally in science you want to validate a result in multiple cell lines because each cell line has its own unique collection of mutations, and if you find something that's true in one cell line, it's possible that maybe that's only the result of some odd mutation that only that cell line has and really isn't representative of that tumor type as a whole. So generally you try to replicate any result in 2 to 3 cell lines in order to be confident that you're seeing something real. This was actually one of the cell lines that I was looking at that didn't work. YAP actually didn't promote brain metastasis in this cell line, so there's really not a lot of brain tumors. So the brain is right there. The one that worked I tested after the image contest. So in these experiments I'm studying human tumor cells in the fish embryo. And by doing these experiments, I'm hoping to learn more about how YAP influences the behavior of human tumor cells. And the hope is that we can learn something that can maybe influence our treatment of human disease.